Welcome to the Arthritis National Research Foundation's Researcher Spotlight webinar series. I'm Laura Douglas, Grant Program Manager, and on behalf of our Board of Directors, Scientific Advisory Board, and staff, I would like to welcome you to the Spotlight on Spondyloarthritis. We are excited to have so many of you joining us from around the world. Before we get started, there are a couple logistical pieces of information we want to share. First, this webinar is being recorded, and we will have it available on our website and YouTube channel in the coming weeks. If you have any questions, please type them into the Q&A box. And if we aren't able to get to yours, please email info@curearthritis.org after the webinar with your question. To get us started, we'd love to hear from you. I'm going to launch a poll, and I want to know what brings you here today. Are you living with spondyloarthritis or an autoimmune disease? Are you a caregiver or a researcher? Be sure to put it in so we can see who's here with us. All right. It looks like most attendees are patients, and that's a great place for us to begin. What we'll learn today, will help us understand what's coming next in terms of diagnosis and treatment. I am now honored to turn the mic over to our patient speaker, Carrie Stiver. Dr. Stiver is the museum administrator, conservator, and executive director of the Buffalo Niagara Heritage Village in Amherst, New York. Carrie will kick off our webinar by sharing her story about living with spondyloarthritis. Carrie.
Thank you so much, Laura, and thank you everyone for having me. As Laura mentioned, I am living with ankylosing spondylitis is my specific diagnosis. So I'm really excited to be able to come here today and share a little bit about my story. I thought that I would just share a little bit about my diagnosis story. I think that it might sound familiar to a lot of you out there because I know that this can be a bit of a tricky condition to be able to get a diagnosis for. And then I wanted to share a little bit about how I've sort of figured out management of the condition along with the role that I have at the museum and other roles that come along with that can be sometimes pretty demanding in terms of time and stress and different activities that I have to be able to do in my sort of day-to-day life. So with that, I'll just get started. So as I mentioned, I am living with ankylosing spondylitis. My first symptoms started around age probably like 24, 25. I am 37 now, so it's been a while. And it started really just with a little bit of joint pain that seemed unusual for someone at that age at the time. Like I said, I was around 24. And I kind of at the time chalked it up to my commute. I had an hour-long one each way commute to my job at the time. And so I thought maybe it was just because I was sitting in the car for so long or, vibrations from being on the road or gripping the wheel really hard. I tend to be a bit of a nervous driver. So I kind of chalked it up to all that for a while. And then other things started popping up, specifically some kind of mysterious bruising that I didn't have an explanation for. So I, at that point, thought I should go get it checked out. So I did, and they ran, all the regular tests, blood tests, etc. And the only thing that came back was that my ANA anti-nuclear antibodies were abnormal. So based on that, I was sent to my first rheumatologist, and pretty much immediately, this first rheumatologist suspected ankylosing spondylitis. But at the time, or just I think that specific doctor, I think there was a little bit more reliance on the HLA marker. So I was tested for that, and it was negative. So the doctor at that time decided that this was not my diagnosis, and we moved on. And so I started off with a diagnosis of fibromyalgia, and was essentially given naproxen and, kind of sent on my way. So things obviously did not improve on that treatment protocol. And so a few doctors and a few years later, I had been through a lupus diagnosis, and then eventually ended up with a rheumatoid arthritis diagnosis. And throughout this time, I was put on hydroxychloroquine. And then really kind of the straw that broke the back, so to speak, was when they decided that for the rheumatoid arthritis diagnosis that didn't seem to be improving, they wanted to start me on methotrexate, which, for those of you who have been on that medication, know it can be a pretty serious medication. And so I was pretty hesitant to do this without feeling confident in the diagnosis that I had. So I was able to find a doctor who was willing to do an MRI, and at that point was able to get my final diagnosis of ankylosing spondylitis. So that was in 2021. So it took 7 years to get my final diagnosis, and like I said, I think this is probably something that can be familiar to a lot of you. Um, so once I got the diagnosis, I started on adalimumab, and that was working pretty well, but I was still having fairly frequent flares. And this was now in 2021, going into 2022. As Laura mentioned in the introduction, I am a museum director. I run a history museum in a suburb of the Buffalo, New York area. At the time, I was also completing my dissertation for my PhD. And so you can imagine, 2021 coming out of the high point of the pandemic, trying to run a museum, trying to do my PhD, and everything else that was going on in the world. There was a lot of stress, and I reached a point where I was having pretty frequent flares, and ending up kind of incapacitated for several days out of the month. And it got to a point where I needed to either, significantly increase my treatment or potentially think about quitting my job at the time. Because my job, of course, because of everything that was going on, was a pretty significant source of stress. And that was not something that I wanted to do. So I was looking for other options, and I knew that physical activity, regular exercise, is a pretty important treatment protocol for ankylosing spondylitis. And I, at the time, was somebody who pretty famously considered myself a person who, hated exercise. I was not athletic. I had never been an athletic, young person. I tried to get out of gym class as much as I possibly could growing up. As an adult, was kind of famous in my family and my friend group for trying different activities and then quitting them immediately. So I had tried fencing, running, horseback riding, gymnastics, all these different things. And I would kind of immediately decide that it was not for me. And even if I had paid for like a 6 week session, I would go to 2 weeks and just kind of be done at that point. So the fact that exercise seemed to be one of the last resorts at this point for me was discouraging. But there was one thing at the time that I had not yet tried that I had some interest in, and that was actually pole fitness, pole dancing. A friend of mine had mentioned this to me as something they had tried. It sounded interesting, sounded kind of fun, and so it was, just kind of the last ditch effort, because I knew that if I wasn't able to find something else that would help, that I would end up having to increase my medication, which at the time was something I wanted to try to avoid. And so I signed up for a pole dancing class, and I went and I completed the 4 week series. And to my great surprise, I signed up for the next series. And then I signed up for the next one, and I signed up for the next one, and I really had found something that I enjoyed. Um, I was so notorious for quitting things when I signed up for them that I actually did not tell my husband that I was doing these classes. because I didn't want him to get frustrated with me for quitting yet another thing. And I was able to kind of do this because he travels a lot for work, so he didn't really notice that I was that I was going to these classes. And it was really about 4 or 5 months later that I went to him and I said, "Hey, I've been doing this thing," and it's been really fun, and I've really enjoyed it. So that was 4 years ago. My 4 year pole dancing anniversary is coming up in October, and I've never been able to stick with anything for this long. And I went from having pretty frequent flares in that first year of doing this activity to having only 3 flares within that first year. So, with my doctor's management and the physical activity, it was something that really was able to help me and has made a really significant difference in how I've been able to manage this condition. So, I didn't quit my job. I'm still here, and things are going, thankfully, very well.
Thank you, Carrie. It's so valuable that you were able to find a fitness routine that really worked to help improve your condition. And thank you so much for sharing your personal experience in living with ankylosing spondylitis. I am now thrilled to turn the mic over to Anna Helena Johnson, who will introduce the diseases we'll be diving into. Dr. Johnson is a rheumatologist and researcher who studies autoimmune diseases. She leads the Johnson Lab at the University of Colorado Anschutz Medical Campus, and her work focuses on certain immune cells, in particular CD8 T cells, and how they contribute to joint inflammation and damage. Dr. Johnson, please take it away.
All right, thank you so much. It's a thrill to be here. I am, as Laura mentioned, a researcher, but today I am wearing my clinical hat. So I see patients in the rheumatology clinic, specifically people with inflammatory arthritis, including spondyloarthritis. And spondyloarthritis is something we're talking about today. A very long word. You're probably looking at it being like, wow, glad you don't have to spell that, right? It did take me a little while when I became a rheumatologist to figure out how to spell this word. Spondyloarthritis is actually the sort of umbrella term for a number of different types of arthritis. And so if you feel like you've been a bit confused about this, don't worry, like it is natural because there's sort of a lot of overlapping terms here. So what I wanted to do is just sort of take a couple of minutes and talk about the different kinds of spondyloarthritis. And then we're going to touch on like very briefly some of the things they have in common before you'll get to hear some really exciting research later on in the session. And so spondyloarthritis is a type of autoimmune inflammatory arthritis, just like rheumatoid arthritis, but the molecules and cells and things that are involved with spondyloarthritis are different. And so this is a different kind of arthritis from rheumatoid arthritis for a number of different reasons. There are many different kinds of spondyloarthritis, and I really like this kind of diagram here with these sort of like overlapping Venn diagrams. You can see up here at the top that it's split into axial kinds over on the left and peripheral kinds on the right. And you're probably like, what does that even mean? So axial here is kind of like the axle on your car. So the axle on your car are the things that connect the wheels. It's kind of like the frame of the car. And axial spondyloarthritis refers to the frame of your body, which means your spine, your pelvis. So axial spondyloarthritis is inflammation of the spine. That can either be what's called non-radiographic, which means it doesn't show up on X-rays, but it shows up on MRI, or radiographic axial spondyloarthritis, which is often called ankylosing spondylitis, as we heard from Dr. Stiver. And so those are both axial types of spondyloarthritis where spine inflammation is the main highlight, anywhere from the neck all the way down to the tailbone, and often including the hips as well. Peripheral means peripheral joints, like your hands, like your feet, knees, elbows, ankles, that kind of thing. Some people have both axial and peripheral, but often people have either axial, either spine or peripheral. And the most common kind of axial or peripheral spondyloarthritis is psoriatic arthritis. So let's see if I can advance the slide here. Psoriatic arthritis is probably the most common of these conditions overall. And it occurs when you have a psoriasis skin rash. And so you can see an example of here on the bottom right of the typical psoriasis rash on the elbow. It can also happen on the scalp. It can happen in people's belly button and your armpit. And it can look in sort of a lot of different ways. but some sort of rash, and arthritis, especially of the hands, but it can also be of the knees, of the ankles, of other joints. And the arthritis of the hands, here you can see that the person's right second digit, their pointer finger, is really, really big. That's called dactylitis, is the fancy name. Sausage digit is the less fancy name. So we see this like particular kind of inflammation that happens in the hands that a rheumatologist can recognize. All right. And so let's see the slides here. There is also undifferentiated peripheral spondyloarthritis. That's a very long term to say, looks just like psoriatic arthritis, but no rash. And so, like we don't have a good name for that yet. And so we call it undifferentiated peripheral spondyloarthritis. The pattern of arthritis is the same and the treatment is the same. And then, as I mentioned, we have axial spondyloarthritis, where we see inflammation where, like here on the lower left, you see a drawing of a pelvis and then the spine fitting into the pelvis. The joints where those things come together, they're about the size of like your two fingers coming together like this. That's called the SI joint, the sacroiliac joint. It moves a tiny bit, to let you like lean from side to side and stuff. That is the classic place for inflammation in axial spondyloarthritis. And so your rheumatologist may have ordered an MRI of your pelvis to see if there's inflammation in those joints because they're so deep that you can't really see them or push on them. And the other thing that happens in axial spondyloarthritis is inflammation at the bones where they meet these discs, these sort of like cushiony things between your vertebrae. And if that inflammation persists, you get fusion of bone growing from one vertebrae to the next, sort of fusing the spine and limiting your mobility so that you can't you can't bend over to tie your shoes anymore. You can't reach like the upper shelves and so forth because your spine is fused. And so this is what we don't want to see anymore in the clinic. And that's sort of the whole goal of the research you're going to be like hearing about today. I also want to mention that in addition to these types of arthritis, there are also other diseases that belong under this bigger umbrella term. So people with spondyloarthritis can also have these other things, including uveitis, which means inflammation of your eyeball, inflammatory bowel disease, and there are two main kinds of that, ulcerative colitis and Crohn's disease. That's when your autoimmune disease also includes your intestines. So your immune system starts to attack your bowels and can lead to diarrhea and blood in your stool and weight loss and things like that. And then some people have just the psoriasis skin rash by itself. Or they can have uveitis and psoriasis or inflammatory bowel disease and psoriasis. It's sort of this mix and match kind of thing of all these conditions. And what brings them together is the mechanisms at play. So there are shared genetic risk factors here. So often in a family, there'll be somebody with psoriasis and somebody with Crohn's disease and somebody with ankylosing spondylitis because the same genes can contribute to each. There's also this cytokine called IL-17 that brings these conditions together. And then we also know that the medications that you can treat all these diseases with, including uveitis and bowel disease and so forth, tend to be similar. And that tells us that the mechanisms behind these diseases is also similar. And so we know quite a bit about these diseases, but there's so much more to learn. And the more that we learn, the more treatment targets we have. And I think that's really going to be the theme of the rest of the session today.
All right. Thank you for sharing that information and for kicking off our discussion and research in such a well-informed way. I want to take a moment to remind attendees that you're welcome to type in any questions you have in the Q&A box at the bottom of your Zoom screen. We are saving them for a great discussion at the end of the session. I'd now like to introduce Dr. Ruth Napier, who will share her research. Dr. Napier is an associate professor at the University of Colorado Anschutz Medical Campus, where her research explores how environmental microbes can trigger autoimmune diseases. By uncovering triggers early, she aims to develop strategies to prevent autoimmune diseases before they start. Dr. Napier, please take it away.
Thank you. It was a pleasure to hear Carrie and Dr. Johnson give their talks. I learned so much from hearing not just the patient's perspective, but also physicians. So now you're going to hear yet a third perspective, which is the researcher. And so I want to start off again just by going over the topic that I study, which is what Dr. Johnson was explaining, which is the ankylosing spondylitis. It's also called axial spondyloarthritis, but today I'm going to call it AS or ankylosing spondylitis just for consistency. So when I first started studying this, I also was shocked by the name. And so I started looking at what it meant. And so it turns out that ankylosing is actually this fusion. It's a process where you actually get fusion of your bones. And spondylitis is any inflammatory disease that affects the spine. So if you're looking down at these pictures on the right, there's a healthy spinal cord. You can see the different vertebrae. And then you can see on the right-hand side, this is a very severe case of ankylosis. And not every patient will get to this stage, but some do. And it's often termed based on this X-ray as bamboo spine. And so as Dr. Johnson was alluding to, the spine actually, the bones grow and actually fuse together. And so as you can see here, it's a literal fusion. It's like cement spine. Which you can imagine is very painful and you can't reverse. So research today is trying to prevent people from getting to this point. And so ankylosing spondylitis is a type of inflammatory arthritis, but of the spine. And as Dr. Johnson said, it's linked to uveitis. About, I believe, 30% of people with ankylosing spondylitis will also develop uveitis, which is ocular inflammation, IBD, and then psoriasis. It's actually fairly prevalent, and now that the diagnostics are becoming more and more clear to physicians, we're getting more and more clear diagnoses, and the rates are going up, but we think it's about 1 in 1,000 people. And while we originally thought the disease was highly proportioned towards males, more recent studies have shown that it's actually maybe a 1-to-1 ratio. It's just that males and females will often present differently. And so don't feel alone if you're a woman. There are lots of them out there. And so what I study is I'm trying to figure out what actually causes ankylosing spondylitis, because if we can figure out what causes it, perhaps we could prevent it or find people prior to getting it to help them seek treatment fast. And so the long answer, or the short answer to the long answer, is that we have no idea what causes ankylosing spondylitis, and there are many ways to get to the same point. But what we do know is that there's a contribution of your genetics in the environment. And so on the right, I have different environmental factors that have been shown in the literature to actually provoke ankylosing spondylitis. Down on the right-hand side is trauma. So bike wrecks, car wrecks can trigger your diet, obesity, pollution in the air, smoking's a big one, and microbes. So this isn't just the microbes that are around us, and this isn't just infections. This is also the microbes within you, like your microbiome. And so if you're combining genetics and the environment, you can see there are many ways in which these two can come together to cause a similar thing. And so then how do we start with what causes this disease? Well, I can tell you how I started. So the first thing that I noticed about this disease is it's a very, very high link to genetics, which most autoimmune diseases, well, there are links genetically, they're not that strong, but in ankylosing spondylitis, it's thought to be like 30%, 20 to 30% related to genetics, which is really high. That means your genetics are a pretty important part of the pathogenesis. The problem is that genetics are very complicated. And so in this country, or amongst Caucasians, we know that about 90% of people will express the protein HLA-B27. And this is not a bad protein. It does not mean you're going to get AS. It just means you have it and that you have a higher likelihood of getting AS. But then there are a series of about 130 genetic mutations, and I'm just listing a few here, like 10 or 15, that actually feed into your genetics. And so that you could have multiple genetic risk factors. And again, as in HLA-B27, these mutations are not actually necessarily bad. So people in the population will have these mutations, and it won't mean that we'll ever go on to getting ankylosing spondylitis. There's some kind of perfect combination of these genetics that will actually trigger it. And it also requires the environment. And so I like to do things in a reductionist system so that I can figure something out. Otherwise, nothing will get figured out. And so I chose to start studying one of the genetic risk factors, which is CARD9. CARD9 is actually a major part of the body's ability to defend itself against fungi. That's not just infections of fungi, but that's the fungi, that the mold that's around us all the time. So you can see in my diagram, individuals are constantly exposed to fungal spores and fungal things, mushrooms in the environment. And that's totally fine because your body expresses card9. And so when cells are triggered, card9 goes up and it mounts a T cell response or a white blood cell response that's associated with secretion of the cytokine IL-17 that protects you against the fungi infection. So you can see on the right, I have the protein card9, and in red, I'm indicating mutations in card9 that actually cause people to lose function of this protein and therefore succumb to brain or meningeal candidiasis. This is very rare. Curiously, there's one mutation in this gene that actually predisposes patients to ankylosing spondylitis and also IBD, which is really interesting. So these two things together made me think that there's something about environmental fungals and this mutation that are triggering ankylosing spondylitis. And so the way I went about testing that is I started with a preclinical model, experimental axial spondyloarthritis in an SKG mouse. And these mice are predisposed to developing axial spondyloarthritis and also peripheral arthritis. So you can see in this picture of this mouse, it's got an ankle, it has a swollen ankle. And so over time, these mice will develop. You can see in this graph here, over time, the normal SKG axial arthritis model, when they get exposed to fungi, they develop arthritis and spondyloarthritis. But when I knocked out the gene, these mice were completely protected. I also showed this using this fancy near-infrared imaging. So you can see in the mice that are lacking card9 that fluorescence intensity goes away, suggesting there's no molecular or clinical presentation of disease in these mice. These data told me that card9 is causing ankylosing spondylitis. So if you go back to the patient mutations, what does that mean? And to sum it up, what we found is that in your blood, so I have a picture here of just your blood, there's white blood cells and there's red blood cells, but a huge contribution of your blood, or 80% of your blood, is actually these purple cells called neutrophils. And so what we found was that these neutrophils in your blood and in tissues, when they're exposed to fungi, card9 expression goes up. And what happens is then these neutrophils get activated and are able to interact with T cells, those cells that we know are important in fighting fungal infections and making IL-17. And for reasons we don't know currently, those T cells are then licensed to go on to the spine and the peripheral joints to cause disease. And so now that we know this access, we're working on ways to interrogate it for treatment and diagnostics. And so this is just a summary of what we showed, kind of distilling down. So we know there's an environmental component or a fungal component, and we also know there's a genetic component, and it likely is more than just card9. It's maybe HLA-B27 or another. Together, they synergize, so you get these pathogenic or disease-causing T cells and neutrophils that cause the development of T cells that then are going to go and attack your spine. And so this is a lot of words, but these are what my lab is now working on. And number one is looking at how we can use card9 and some different findings that we have associated with as a disease biomarker of ankylosing spondylitis for helping with diagnostics. And also knowing if you have this mutation, would you be more susceptible to uveitis, arthritis, or IBD? We also are looking at it in terms of being a predictive biomarker of treatment responders. So when you come into the clinic and you're diagnosed with axial spondyloarthritis, there are multiple treatments that are FDA approved, and the physician at the current state does not know which drug is going to work in which patient. So it's somewhat of a trial and error system, and we're hoping by using something like this marker that we can pull patients out of the clinic knowing right when they get diagnosed that they would be responsive to certain treatments like IL-17 inhibition. And finally is just we're trying to reveal new cellular targets to develop drugs for the long run to fix underlying diseases. And with that, I will thank my lab and my collaborator. And Dr. Johnson and I are actually in the same division at CU Anschutz. And last month, our division won the costume contest at the annual rheumatology retreat or immunology retreat. And this is us in Jurassic Park. I'm in the T-Rex costume. And I just want to thank my lab members, especially Emily Vance, Leah, Amy, and Kristen, and my collaborators, Dr. Laron Kaplan, Holly, and Atul, and my funding from the ANRF. Thank you.
Thank you so much, Dr. Napier. The work you're doing sounds incredibly promising for people living with spondyloarthritis. I again want to take a moment to remind everyone that you are welcome to type any questions into the Q&A box at the bottom of the Zoom screen. I'd now like to introduce Dr. Jimin Tan, who will talk to us about how he uses AI to study genomics, which is the study of DNA, in order to better understand how the autoimmune diseases like psoriatic arthritis function. Dr. Tan currently holds a postdoctoral fellowship and is a Colton AI scholar at NYU School of Medicine. He has a PhD in computational biology, and coming from a background of mathematics and computer science, he aims to develop machine learning methods and models to facilitate understanding of autoimmune disease. Dr. Tan.
Thank you, Laura, for introducing me. And it's a great pleasure to be presenting here and also hearing great talks from Dr. Napier. And also, it was a great introduction by Dr. Johnson about psoriatic arthritis. Actually, I have my first slide, but I guess I can skip most of it. So, yeah, psoriasis and psoriatic arthritis are common autoimmune conditions, and one of the most challenging tasks in the field today is to understand how does a psoriasis patient progress from psoriasis to psoriatic arthritis. So there are 20 or 30% of patient does that, but the underlying cause is still remains elusive. So if we let's see. The slides is okay, great. So if we sort of look at this problem on a very high level, if we have some sort of disease, there's usually a molecular and biological underpinning behind it. This usually means that our tissue or cells has gone from a normal state to a disease state. So in order for us to answer this question and understand sort of why we have progress from psoriasis to psoriatic arthritis, we need to compare the two different cell types, one from normal and one in the psoriatic arthritis condition. So so this is where genomics comes in. Basically, genomics gives us a set of tools and perspectives that we can look inside of the cell and examine different molecular mechanisms that driven cellular functions. And so usually this cellular function that goes wrong and causes disease. So if we can leverage our genomics tool and of course combine with machine learning, this will give us a better understanding of disease mechanism and help us to develop treatment for disease conditions. So again, going back to the disease to cellular state changes, if we have a disease that sort of deviate from normal state, this usually means that the cell or tissue will have different protein expression level because protein are sort of the building blocks of cellular structures. So, and if you still remember courses from high school biology, the central dogma of biology implies that there are some sort of regulatory changes for RNA expressions and translational differences in protein, and that leads to the protein differences. And actually, transcription is one of the most complicated mechanisms in the cell to make sure that the cell is functional normally and producing the right amount of proteins. So it is a really, really complex system. You can almost think about different components in the regulatory system as cogs in a huge machine. So you can think if just some cogs goes wrong, then the whole machine might break down. So by understanding the regulatory mechanism of many diseases, people have been able to produce drugs that cure this disease. This can include melanoma, many autoimmune conditions, and here also HER2 breast cancer, which is a subtype. For many of these, we know the mechanism, we can develop drug to treat them However, there's still many abnormal conditions and diseases that we don't know the mechanism, and therefore we cannot, well, this jumped too fast. We cannot sort of develop an effective treatment against them. So this includes one of them is T cell exhaustion, which I'll be talking about today, and then also many autoimmune conditions, including psoriatic arthritis. Sorry, there's a bit of a lag. Denzel, could you, is it, oh, okay, great. Uh, yeah, to put all of this into context, I want to showcase a case study. This is about T cell exhaustion in the tumor microenvironment. Although this is not directly autoimmune disease, but this is usually thought of sort of the opposite side of autoimmune disease. This is also on the immune cell, but rather than inflammation, this actually suppresses T cell inflammatory function. So CD8 T cell are a type of cell that kills infected cells or malignant cells, basically like a fighter in the human immune system. And when encountering stimulation, it gets activated and becomes activated T cell that is sort of exert its killing functions. However, due to during prolonged stimulation, chronic stimulation, this T cell actually gets into an exhausted state. This is a protection mechanism to prevent overactivity that will damage our organ during like a long-term infection that the immune system cannot clear out. So in order to for us to study this process, we first need to compare what is called an infected T cell, which are essentially activated T cell, against exhausted T cell. So we need to compare between these two cell types and understand what exactly is going on in the regulatory mechanism behind sort of underlying this cellular transition to understand the process and then design therapeutics or other intervention mechanism to sort of revert or even or prevent it. So as we talked about before, the central dogma of biology, many of this actually happens in the transcriptional level. So one thing I want to somehow cannot advance the slide. Okay. Oh, that's too much. Oh, yes, great. Thank you. So a very key part in our regulation process is a set of protein that is called DNA binding protein. So these are protein that binds to the DNA and regulates our gene expression. So we have 30,000 different genes, and our body, in order to function normally, we need to express different genes at the adequate level. And you can imagine if some gene goes out of order, then the cell will go into an abnormal state, which can lead to disease. Okay, can we go to the next slide? Thanks. Yeah, so these DNA binding proteins looks like the graph on the right side. These are small boxes, and they basically goes into contact with our DNA sequence. And on the right side, you can look at how different proteins binds differently to different locations. So different locations sort of regulates different type of genes. So it's this sort of combination of different type of DNA binding proteins and their specificity to different genes that allows us to have consistent expression of different type of genes. So in the human, there are around 2,500 DNA binding proteins, which is a lot. So in order to maintain a stable level of expression, the cell really develop a huge tool to maintain it. However, this brings the complexity that in order to understand the whole picture of regulation, we need to profile over 2,500 proteins, which is a great challenge. Can we go to the next slide, please? Yeah, so imagine going back to the exhaustion versus the effector immune cell problem. Imagine we can actually measure the DNA binding protein level for these two different cell types. And ideally, we want to find the protein that have differential binding that will introduce differential regulation between these two cell types. Can we click that again? Thank you. Yes. So, however, we need to do this across all 2,500 proteins. So this is a really arduous and long process. And so people have tried this before. Oh, I think we're going to, yeah. So, oh, can we go to the last slides? Sorry. Yes. So in order to measure this, this is a really long process because there's a lot of data, and the current assay we have can only measure one protein at a time. So in order to measure the entire 2,500 protein, it actually took people more than 5 years to complete the whole measurement in one single cell type. However, for our application, we need to measure this across two different cell types because we want to know what's going on and what's different between them. So it is really impossible for us to perform this experiment in the lab. And that's why we resort to machine learning to solve this problem. Can we go to the next slide, please? And further, yes. Okay. So usually, normally, if we were to measure this experimental data in the lab, it would take more than 10 years. However, use our machine learning model, we can speed it up more than 10,000 times. And actually, the whole prediction for these two, for this rich functional data now takes a little under 2 hours. So this is how machine learning can actually be used to speed up our research. And this is our model. On the left side, this is our model architecture. We take different type of genomic input to make sure that we're capturing the correct binding signals and make accurate, also making accurate predictions. And on the right side, you can see this is our actual prediction, and the top track is our prediction, and then the light track on the bottom is the ground truth protein binding profile. And you can see our prediction greatly sort of mimics the experimental ground truth, which is great. And I didn't put it in here, but this model also outperforms most of the state-of-the-art models. So it's the best model so far. So we know that model performs really well. Now we go back and reapply this model to study the difference between exhausted T cells and effector T cells. And by pumping input information into our model, we're able to generate this rich functional data that would otherwise take more than 10 years to measure. And so by analyzing this data, we actually ended up finding actually many of the transcription factors, regulators that people have previously identified. If you look at the right side here, we see that BHL-HE40, KDM6A, which is both discovered in 2019, 2021. But the T cell exhaustion field sort of started in the early 2000s. So it took researchers over 20 years to find all these markers. But with our model, we can do them in under 1 day, which is a great efficiency increase. And other than identifying markers that people previously have found, we also find a novel protein, which is ZNF766, that hadn't been previously characterized. So we went down and performed validation on this new protein that we find. So what we did is that we in the T cell we remove this protein, and we see now if the T cell has more function, is less prone to exhaustion and more active. And in fact, we actually see there's a great reduction in exhaustion marker, and also a great increase in cytokines. This means that in the absence of this protein, T cell is now more functionally active. So this is really game changing, because now previously you have 2,500, but you don't know which one is, or which ones are the ones that regulate this process. But now through machine learning, you can complete this screening in a very short amount of time and identify really the drivers of the disease. And then once you identify this protein, we can design therapeutics against this protein to sort of prevent as a prevention and intervention mechanism. So right now, we are developing a new research program where we use our model to study psoriatic arthritis. So in this study, we're most interested in identify what is the difference between the normal individual patient with psoriasis and patient with psoriatic arthritis. So we have now successfully collected single cell data from both normal individual and psoriasis patient, and we're currently in the process of collecting data for psoriatic arthritis. And so the hope is that we can run a similar analysis with this model framework to compare between psoriasis and psoriatic arthritis and identify really the driver protein that drive the disease progression process. And the idea is that once we find these proteins, we can look upstream pathways and search for existing drugs, and that will give us new therapeutic molecules to prevent progression of psoriatic arthritis. And with that, I want to thank all my collaborators and ANRF funding. Thank you.
All right, thank you, Dr. Tan. Before we open it up for Q&A, we'd love to hear from you. We know rheumatic diseases impact a lot of people in many different ways, and we'd like to know what's the number one thing you'd like to better understand. You'll see a quick poll on the screen with a few options. Go ahead and choose one that resonates most with you. Is it symptoms and flare patterns, diagnostics and testing, research in the future? Which of these options do you see that you feel you're most interested in? All right, thank you for sharing. That's super helpful to see. We've got questions coming in that touch on some of these different areas, and we'd love to dive into Q&A from our speakers. So I'll ask my panel to come off mute if you don't mind. And we'll start with question one, which is for our researchers. Many people with psoriatic arthritis and axial spondylitis experience delays in diagnosis. What progress is being made to help them be diagnosed earlier?
I guess I could start. I think there's two things. One is physician education on diagnosis. I think it's not that people are behind the boat, it's that these guidelines are being actively designed by experts in the field, of which there's a growing capacity of, but there hasn't historically been a lot of axial spondyloarthritis experts. And so I think what they're having now is they're hosting like clinics and being more showing people how to like read x-rays, because a lot of time it can get mixed or missed, and people don't know exactly the tests to do. And then I think the exciting one for diagnosis is there's a lot of work being done in like the biomarker genetic space, with polygenetic risk scores being able to predict disease. But I think the field is moving towards personalized medicine where your personal genetics, like the stuff I do and other people, will inform treatment strategies.
Really great. Anyone else?
Yeah, I agree with what Dr. Napier said. Like as a rheumatologist, I think it's sometimes I think it's much easier to get an MRI now than it was even 10 years ago, and definitely 20 years ago. And I think that definitely is going to make it easier to get diagnosed. The other thing that we're working on right now is having people realize that many women get this. I think right now a lot of traditional rheumatologists kind of have their like blinders on, and they think like ankylosing spondylitis only happens in like young men, younger than like 25. But in reality, you see it in a much broader age range. It's much more equal in terms of genders. It happens in people who do not have HLA-B27, and so there are certain um, uh, preconceptions that have turned out to be incorrect that we're trying to overcome. And so I think that's the progress that's happening right now. I think ankylosing spondylitis is due for one of those like next generation science kind of things with like, could there be a more complex genetic test that we could send on patients from their blood, that would help say like this person is at high genetic risk of ankylosing spondylitis or at lower genetic risk. But we don't have that yet. That gets very very tricky because again, there's not one gene that people have. It's probably like 15 or 20. And lots of people have like 10 of those and are just fine. And so it's a complex test to design.
That's great. Thank you, Helena. Our second question is for Carrie. Many patients wonder how to advocate for themselves at appointments. What's worked for you?
Yeah, it can be difficult. I think the single biggest thing that was most helpful for me was documenting and making sure that I told my doctors everything. At one point, I had a doctor who said to me that I am a good historian um, of my own condition, which was funny because I am a historian in my job. So maybe this is where this comes from. But really documenting my symptoms, my medications, things that seemed to trigger symptoms, but I think the thing that really ended up getting me to the most helpful diagnosis and the most helpful place was documenting the fact that the treatments that they had me on really didn't seem to be working. So I know it sounds like a lot, and it kind of is, but really working with your doctors to make sure that you have as much information as possible.
That's great. Thank you, Carrie. We have a question that's a little more specific to doctors. So, Helena, if X-rays and MRIs don't show damage or inflammation, but someone still has widespread pain along with psoriasis, how do doctors determine whether it's psoriatic arthritis or axial spondylosis?
Yeah, this can be really tricky because there are lots of things that can cause back pain, not just inflammation. So the most common cause of back pain is, what we often call like lumbago, right? When you have sort of a muscle spasm or sort of that's the sort of like thing that happens in sitcoms and things like that, where people throw out their back and then they're stuck on the couch for a couple days and things, and you can get milder versions of that. And the thing is, if you have psoriasis, the most common cause of back pain is going to be that kind of back pain because it's the most common overall. And so I think that I'm really grateful that MRIs exist because they help to diagnose inflammation. And so if you have back pain, but your MRI does not show inflammation, it's unlikely that you have ankylosing spondylitis. And so like I would say give the diagnosis that your physician is offering to you a chance. If they're saying like it's a herniated disc or some sort of other thing and they're recommending physical therapy, like give that a shot. And then as Dr. Stiver said, like document, like if you try that thing in good faith and it does not help, that is another piece of information that your doctor can use. But don't like non-inflammatory types of back pain and joint pain happen in people with psoriasis. They happen in people with Crohn's disease. They happen in people with all these other things. And so don't it doesn't necessarily mean you have inflammatory back pain if you have back pain.
Great. Thank you. All right. And looking ahead, what are you most excited about for the spondyloarthritis community? Could it be what's on the horizon for research or patient care or support?
I think I can.
Oh, go ahead. Go ahead, Jim.
Okay. Yeah, so I think for early diagnosis, there could be a possibility of using, for example, smart watches or smart rings. Maybe there are some biometrics that can be picked up in those devices and use, across time tracking with machine learning algorithms. Maybe there's an increased accuracy in detecting these, symptoms early. And then in terms of treatment, I'm really a believer in understanding the genomics using large scale machine learning tools because that can actually enable you to get, for example, like a small maybe biopsy of the uh, uh, like the disease region, and then performing relatively low throughput experiments, but then run really high throughput machine learning, high capacity models to understanding, like sort of what is the patient specific cause, or maybe there's some variants that might require a little bit different treatment, right? So I think the future of precision medicine and precision treatment for spondyloarthritis is really exciting to me.
Great. Anyone else?
I mean, I was just gonna say that I'm really excited about like how much attention has been put on spondyloarthritis in the last maybe 5 years. And I think kind of what Dr. Johnson was showing is how all the different conditions overlap, like genetically and clinically, that now we're able to like push all of these great biologics into the spondy field. And I think we're really close to knowing like who will respond to what. And I think if we can boost treatment, which I think we're headed in a good direction, like diagnostic and treatment, and who like people getting diagnosed and getting put on treatment. And if we can get people on the correct treatment, I think we'll see better outcomes. And I don't know, it just excites me that I think more people are starting to put their heads together on this, and I think it's heading in a good direction. So I'm optimistic.
I think if I were to give what I'm most excited about is I think that in the last 5 or 10 years, new technologies like single cell RNA sequencing and other sort of fancy research tools have revealed like totally new avenues to treatments. So Dr. Napier talked about one like neutrophils. I don't think anyone has really thought about neutrophils and how to target them in ankylosing spondylitis. And so that is sort of the beginning of a path towards a completely new treatment family, because I think that's, as a rheumatologist, what I worry about the most is, right now we have only like 2 or 3 classes of medications that work for ankylosing spondylitis. And so if one stops working and then you have an allergy towards another or something like that, like the choices get very slim. And so I'd like to sort of beef that up so that we have more options. And I see that on the horizon.
That's fantastic. Well, thank you all. I'm getting word we are approaching time here. As we wrap up, I want to say thank you so much to our fantastic patient speaker, Carrie. We are so appreciative of you being here to share your story. Also to our wonderful researchers, Helena, Ruth, Jimin, for sharing your innovative work towards understanding spondyloarthritis. You can see firsthand the impact ANRF has in supporting these incredible individuals in their quest for a cure. Thank you to everyone who's joined us to make this event great. If you enjoyed this webinar, we kindly ask you consider making a donation today by visiting curearthritis.org/donate. Every contribution helps. We will be sending our live attendees a survey after the event. Your feedback helps us find direction for future webinars. Please share your comments with us so we can continue providing important and relevant content to you. And please don't forget to sign up for our other Spotlight Series webinars. We will be live at the same time on October 8th with Sjogren's disease and October 22nd with rheumatoid arthritis. Thank you for coming, and please remember to follow us on social media to stay well connected.